another in the cell. So let's start with post-translational processing, and the first thing we'll talk about is this modification process. And there are different types of modification, as you might have guessed. The first thing that can happen in terms of protein modification is that sometimes a piece of the brand new polypeptide will be removed. And there are two ways that this can happen. One is called cleavage, and the other is called splicing. And the only difference between these two is whether or not you're talking about a portion on the ends of the polypeptide being removed, or a portion in the middle of the polypeptide being removed. If it's removed from the ends of the new polypeptide, that's what we call cleavage. And if it's removed from the middle, if a section of the amino acids are removed from the middle, then the remaining sections have to be joined together, and that's what we call splicing. And that's very similar to what we saw for mRNA processing, which also involves splicing in eukaryotes. So, in terms of modification, when we talk about bacteria, again, we're talking about a simpler process in terms of modification. And the first thing that happens in bacterial proteins is that the start codon amino acid is going to be removed. So the start codon, remember, is AUG. That's the sequence of 3 bases that's on the mRNA that tells the ribosome that this is the place to begin translation. And those 3 letters combined in a codon, AUG, signal the tRNA to place a methionine there in the first position of every protein. And of course that corresponds with what we would call the N terminus, the amino terminus. So that methionine gets cleaved off. It gets removed from the brand new protein in our bacteria. Now, the other thing that can happen is additional amino acids, whole blocks of them, can be removed in order to activate or turn on this new protein. And again, if that happens at the end of or at one of the ends of this brand new polypeptide, we would call that cleavage. Some proteins have internal sequences of amino acids removed, and that's what we call splicing. One of the best examples, or a well understood example of modification involving splicing, is in our hormone called insulin, and I'll talk a little bit more about that in a minute. Now, proteins that are going to be transported across a membrane will have to have a block of amino acids removed from them that are called signal sequences. Now, signal sequences are at one end of the brand new protein, and so they're going to be cleaved in order to remove them. And we're going to talk much more about signal sequences in a few minutes. So, protein splicing, removing an internal sequence, is very analogous to the process of splicing in mRNA. Remember, in mRNA, the introns get removed so that the exons can be joined together to make the coding portion of the mRNA. In proteins, we use similar terminology. Instead of introns and exons, we talk about inteins and exteins. So inteins get removed from proteins, and exteins get bound together. So here's a question for you. What kind of an enzyme breaks a peptide bond? Remember, a peptide bond is a type of covalent bond, and that means it's going to take some work to break it. If you don't know the answer to this question, see if you can find it. See if you can find it, and we'll talk about it in class. The name of the group of enzymes, the general name of the group of enzymes that can break protein bonds. So this is a diagram that comes out of your book that just diagrams for us these processes of protein modification, including cleavage, and splicing. So if you look at the very top of the diagram, you can see this precursor protein, this brand new polypeptide that's been translated and set free. And this one has an interesting interaction forming already called a disulfide bond. Remember, this thing is a chain of amino acids, and so it's going to have R groups that are going to be interacting with each other, chemically attracted or repulsed from each other. And in this case, what's happening is we have a cleavage event where the brown portion of the protein is being removed, and the final protein then is shown in purple. Now, protein splicing in this picture, in the middle picture, is what we would call protein splicing. This is intramolecular. In other words, it's within one molecule. We didn't talk about this intermolecular process, so don't worry about this bottom portion. But what you have over here is a precursor polypeptide with green, yellow, and purple regions. And it's the yellow region that represents, excuse me, an intine, or a portion of a protein that's going to be removed. Then the green and the purple portions, the extines, will be spliced together to make the final protein. So, in protein modification, cleavage and splicing are the first things that can happen with a protein. The second thing that happens is referred to as chemical modifications, because this is an instance where something's going to be added onto the amino acids at some location. There are three main molecules that get added on to amino acids, and then there's also a process where carbohydrate side chains can be added to a protein. We've talked about this a little bit before in past lectures. This is a process called glycosylation. Remember, when you see that term glyco, that's usually referring to carbohydrate. So sometimes we're going to see a carbohydrate side chain added onto a protein, and that will turn it into a glycoprotein. For our small molecules, we're primarily talking about methyl groups, CH3, phosphate groups, PO4, and acetyl groups, COCH3. So methylation, phosphorylation, and acetylation are our three typical chemical modifications, in addition to glycosylation. In this slide, you can see a representation of the amino acid lysine on the left-hand side. So what they're showing you is that where the LYS is the central carbon and the amino group and the carboxyl group and the single hydrogen atom. And the R group is what you see here with this broken chain of, or this bent chain of carbons and hydrogens, and then this amino group at the end. So this is what we would normally expect as an R group coming off of lysine. Now we can modify that and turn this into acetyl lysine by adding this accessory group up here, this acetyl group. We can also modify it and turn it into trimethyl lysine by adding these accessory methyl groups. And that's what we would refer to as chemical modification. Now this is going to make a difference to how this protein, this amino acid within this protein, behaves. Because once we alter the exposed R group, we alter its chemistry. So this original R group would behave one way, and now we've added an oxygen and a methyl group, and over here we've added 3 methyl groups. So we're going to change the chemistry of that amino acid by changing its R group, modifying its R group. The last thing I wanted to talk about in terms of modification of proteins is this process where sometimes multiple proteins can be bound together to create, again, quaternary structure. and what we would call a multimeric protein, a protein that functions as one, but is actually composed of multiple separate proteins. And again, what we refer to this as is a multimeric protein. One protein, functionally speaking, but it's composed of a blue and a red and a green and a yellow protein joined together. So in this slide, what we have is sort of a summary of what's going on so far in terms of our protein processing. Up at the top of the slide, you can see a representation of the DNA double helix, and they're showing us that we've got a coding sequence here, or what would be referred to as an exon. We've got an intervening sequence, or an intron, and then we've got a coding sequence on the end, another exon. And remember, the RNA that gets made from that DNA is going to have all those components in it. And this intervening sequence, this intron, can be removed here when the RNA is processed. Or, alternatively, and in the case of certain proteins, like the insulin protein, what will happen instead is that at the protein level, we will remove the new intine and join together the resulting extine. Extines. That's a procedure or a process that's called splicing because it involves an internal region of the protein. The red intine gets removed and the remaining green extines get joined together to produce a mature protein. And again, insulin is a nice example of this. We actually have studied insulin a great deal. We know how insulin is produced. We know what insulin does. And it turns out that insulin has two levels of modification. It starts off as what we call pre-pro insulin. Pre-pro insulin. So it's got two stages before it becomes insulin. What you're seeing on the left-hand side of the slide is a representation of this immature protein. Here's the amino group, or here's the N-terminus. It's got a signal peptide in it. That's a portion that's going to help it go where it needs to go in the cell. It's got a region that gets called the beta chain, and that's not terribly important for us to understand, but it's got a loop of amino acids followed by what's called an A chain. So from B all the way through C and into A is just a chain of amino acids leading to the C terminus, or the carboxyl group. This is what we call pre-pro insulin. Now once the signal peptide takes the pre-pro insulin where it needs to go, that signal peptide gets removed. So that's a cleavage event. And as you can see over here in the middle, the signal peptide is gone. Now we have what we would call proinsulin. It's not insulin yet, it still needs some modification, but the modification has begun with cleavage. Now in this central image, what they're showing us is that the B chain of amino acids and the A chain of amino acids are going to begin interacting with each other. They're going to begin folding with these disulfide bonds. That's what's being represented by SS here and SS here, disulfide bonds. So we have some chemical interaction going on here, but we're still not near where we need to be to have functional insulin. That doesn't happen until we remove the central loop of amino acids labeled C chain. So that's a process of splicing an intine and joining extines, and that's what you're seeing over here. Over here we have the final insulin molecule. It's got an N-terminus and a C-terminus for the B chain, an N-terminus and a C-terminus for the A chain, and multiple disulfide bonds holding it together. So all of that together is an example of a molecule that goes through some extensive protein modification. Now, once the young protein, is modified through cleavage and splicing and chemical modifications, then it can start into this process of targeting and sorting. Now, remember, when we talk about targeting, we're talking about proteins that need to go to a specific destination, and that happens using a signal sequence. So just like our pre-pro insulin had a signal sequence on it, lots of proteins have a signal sequence at the N terminus in order to get them where they need to go. Now we're going to break this part of this material into two sections. We're going to talk about co-translational import and post-translational import. That term import refers to moving the protein into, or importing, into some other structure in the cell. So for co-translational import, we're talking about proteins that are destined for the Golgi apparatus, the various transport vesicles, or what are sometimes called endosomes in the cell, lysosomes in the cell, or to the plasma membrane. The other way that co-translational import can be used is to get proteins that are ultimately destined to go to the ER. In other words, if the final destination for a protein is the ER, it has to move through other organelles first and then basically go back to the ER. Same thing for the Golgi. You'll see proteins that get passed through co-translational import through the ER, through the Golgi, and then back to the Golgi, which is their final destination. So we'll start off talking about co-translational import, and then we'll talk about post-translational import, which is a process for proteins that are destined, ultimately, to end up either in the cytosol, in the nucleus, in the mitochondria or the chloroplast, or into the peroxisome. And this is a summary slide of those various processes, and this will be a good place for us to end today. This is a diagram that comes right out of your book, and I really like it because it puts in enough detail to help us differentiate between co-translational import and post-translational import. Remember, those two terms refer to this process of trafficking and sorting depending on where the final destination is. Now, up at the top of the slide, they're showing us the mRNA interacting with ribosomes to make that original immature polypeptide. But that polypeptide then has to have a destination where it's going to finally end up. And depending on that destination, it's either going to go through co-translational import or post-translational import. On the left-hand side of the screen, you can see the mRNA and the ribosomes translating it, and it's being imported first into the endoplasmic reticulum, shown in blue. It's then going to leave that endoplasmic reticulum, go into the Golgi, or it might go right back to the ER if its final destination is the ER. Once it's in the Golgi, it's going to be processed further, and then it's either going to come out in these little transport vesicles and end up in the membrane. Perhaps it'll end up in lysosomes, or it may end up in a secretory vesicle. In other words, in a membrane-bound compartment that's going to take it to the membrane and release it to the outside of the cell. On this side, we see something very different with post-translational import. We're missing the endoplasmic reticulum. We're missing the Golgi apparatus. These are proteins that are destined for the nucleus, or the mitochondria, or the chloroplast, or the peroxisomes. So we still, of course, see translation occurring at the level of the ribosomes. We still see proteins being modified and folded, but this time they're skipping the ER and the Golgi complex and getting packaged instead directly, imported directly into their various destinations. So we're going to talk about each one of these separately, and that's a good place for us to end today.